Hello AP Bio, this is Mrs. Kasperwitz, and in this quick video notes I am going to be reviewing with you the differences between prokaryotic and eukaryotic DNA. So in this picture here we have a picture of prokaryotic DNA. So just remember that a prokaryote is a cell that does not have organelles to it, right? It's a very simple cell. It'll have a cell membrane, possibly a cell wall. It will have a piece of circular DNA um, and ribosomes and cytoplasm, but it does not have the mitochondria and the chloroplasts and the ER and the lysosomes, all of those things that we associate with eukaryotic cells like plant and animal cells. So prokaryotic cells, general thing, no membrane-bound organelles, including no nucleus. Eukaryotic cells are going to have all of those membrane-bound structures in addition to a nucleus, which will house their genetic information. So we're going to start with some of the similarities between prokaryotic and eukaryotic DNA. So first of all, the actual DNA itself, all right, in all cells, prokaryote to eukaryote, is going to be that double helix, okay? So and by what I mean by similar 3D organization is all of those calculations that Rosalind Franklin did. For example, the distance between the coils or the twists, the distance across the front here, okay? What's the distance between the different nucleotide bases in the ladder? All of those 3D organization things of the helix itself are similar across all cells, prokaryote or eukaryote. They follow Chargaff's rules, all right? So the proportions of A's and T's will be 1-to-1, and the ratio of C's and G's will be 1-to-1. And they will also follow the base pairing rule, which is that A pairs with T and C pairs with G when we're making the double helix, the nitrogen bases pairing rule. The strands, like we've talked about before, are going to run anti-parallel to each other, right? So one strand is going to be running in the 5' to 3' direction, and on the opposite side, the complementary strand is going to be running 3' to 5'. Right, so that's true again across the board, all prokaryotes and eukaryotes. As we saw today, replication occurs in a similar manner. There is the little difference between the prokaryotic replication in that there's just one site of initiation or one origin, whereas in the linear pieces of DNA that we find in a eukaryote, you're going to have multiple locations of origins to begin the replication process. But other than that, the leading strand, the lagging strand, the Okazaki fragments, the enzymes involved are similar, right? Both prokaryotes and eukaryotes will have what we refer to as extra chromosomal DNA. In a eukaryote, we sometimes refer to that as non-nuclear DNA because our DNA is encompassed in a nucleus, but extra chromosomal just means not the main chromosome. So in a prokaryote, as we'll see in just a minute, they will have extra pieces of DNA called plasmids that are separate from their main genome. And in eukaryotic cells, we can have the extra chromosomal DNA would be in the mitochondria and or in the chloroplasts if you're a plant cell. I believe that some eukaryotic cells can also take up plasmids, all right, which would be in the cytoplasm then of those cells. So in a prokaryote, we have here a picture of a plasmid that was taken from E. coli. And this particular plasmid is famous because it was the plasmid that they used in the first genetic engineering experiments done by Cohen and Boyer. So what they did with this plasmid is they introduced into this piece of bacterial plasmid this extracellular piece of chromosome that the bacteria E. coli have. They spliced into it a gene from another organism. So the bacteria cell DNA, this plasmid DNA in the bacteria cell, was genetically engineered to include a piece of DNA from a completely separate species, right? And they were able to have bacteria cells then uptake this plasmid, carrying this new gene that was not bacterial in nature, and that plasmid was able to then alter the phenotype of the bacteria cell in which it resided. Um, in nature, the plasmids can be transferred from one bacteria cell to another in a process called horizontal gene transfer. This is how bacteria cells within the same generation can share DNA and increase genetic variability in their cells. Um, so here's a little bit different picture here of it. So here would be our main chromosome, our main circular chromosome in the bacteria cell. And then we've got plasmids, which is extracellular DNA, these little tiny circular pieces of DNA. Um, these plasmids are very interesting. They can carry genes for antibiotic resistance and transfer that antibiotic resistance from one bacteria to another in the same generation. These plasmids can be inherited, all right, by the next generation of bacteria that come in a vertical gene transfer, which is the normal reproductive process, right? Um, so if you'd like to know a little bit more about these plasmids, definitely click on this picture to learn a little bit more. We are going to be talking about them and using them in our genetic engineering labs when we get back from the break. So now, what are some of the differences between the prokaryotic and eukaryotic genome? So in the prokaryotes, in the bacteria and the archaea, the DNA is found freely floating around in the cytoplasm. It's not encased, as we said, in a nucleus. It's not membrane bound. They do have a region that they call the nucleoid, but it's, again, not surrounded by a membrane. Also important here, and I think this is why the replication process can happen more quickly, as can the translation and transcription of the prokaryotic DNA, is that we say the DNA is naked, meaning that it's not wrapped around these proteins that we call histones. All right, so because it's fairly simple and it's a circular piece, it's not bound up with these histone proteins like we find in eukaryotic DNA. The genomes are compact, which basically means that there's very little repetition of DNA sequences that we would find in a eukaryotic cell, and there are no non-coding sequences that we refer to as introns. Okay. By that I mean there would be the DNA of a prokaryote codes for traits. All right, so there is no parts of that DNA in a prokaryote that are not responsible or are not genes that code for particular traits. We already said that they contain these extracellular plasmids, and again, they're circular in shape. So eukaryotic DNA, there's a lot more of it, right? We enclose it inside of a nucleus, all right? So it's membrane bound, it's within a membrane bound structure. And it is associated with proteins called histones, right? And I'm going to show you a picture of those in just a second here. But basically, the DNA double helix gets wrapped around these proteins called histones, right? And that's going to be one of the levels of organization that we find in eukaryotic DNA, all right? Our genome contains large amounts or large sections of base pairs that don't code for any particular trait, right? And we also have large sequences of DNA that is repetitive in nature, meaning that we'll have the sequence CCGG, 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 and it'll just keep going. Okay. We also those repetitive and non-coding sections include introns, which are, again, are segments of DNA that do not code for a particular protein. Again, I'm not sure about this, but I've read two conflicting things. So right now I know for sure that we have non-nuclear DNA in the mitochondria and in the chloroplast if you're a plant, in addition to the mitochondria in a plant. I've read some sources where we are finding that eukaryotic cells can also contain plasmids, but I don't want to be quoted on that. All right. And they are linear in shape. So they're long, long things that look like a string. Okay. So because we have so much DNA, all right, in a one single piece of chromosome or one piece of DNA, we have to organize it. All right. So in eukaryotes, there are levels of organization in our DNA. So you start with the double helix. Okay. And then we do something called supercoiling. All right. So inside, these proteins right here on the inside are called histones. All right. And the DNA, the double helix, gets wrapped kind of like a spool of thread. So you imagine the histone would be the spool of thread, and then the DNA is the thread around that spool, that protein. Okay. That's how you can imagine this level here. And that forms a structure called a nucleosome. So this histone protein with the DNA that's been wrapped around it is referred to as a nucleosome. And then that coils into something we know of as chromatin, which is the form that the DNA takes in a non-dividing cell during interphase, okay, in the G1, right? So if we look over here at this picture here, you can see this level, okay? This one right here is in this picture. So each of these little things that looks like a bead on the string is one of those nucleosomes. All right, those nucleosomes then coil up some more, okay, and eventually we get to what we know as the chromosome form, which is the highly condensed, supercoiled pieces of DNA that we can see under the microscope and follow through a cell division, either mitosis or meiosis. Okay. So if we look at that packaging now, we can see here, this would be a cell that's undergoing a division now because the DNA has been copied, right? So we've got a chromosome with its sister chromatids attached to each other. So here would be my centromere, and this would be the highly coiled version of the DNA. So please click on this picture at your leisure and watch a short video on the packaging of DNA. It shows you in an animation the coiling process, which is very hard to describe in words. It's much better to see in person. So I think the video clip's about 2 minutes long. So that's some information that you might want to have in just some background knowledge about the differences between prokaryote and eukaryote DNA and how it's packaged within the cell and how it's put together. Okay, so please do watch the videos here. They're both short clips that will talk to you a little bit more about the plasmids and also about the structure of the eukaryotic chromosome. Thanks and have a good rest of your day.